Hello, welcome to Tattoo Universal. This is A-level CIE Biology, and right now we are on to All Topics Series 3. So we are solving all topics. So you see questions from different topics in this particular class. Okay, this is schedule. Let's go to number 1. So get your materials, your writing pad, your pen. Let's dive in. Okay, let's dive in. Let's go. Now, which role describes some properties of stem cells? Okay, now it's asking us, pause this video and try to answer it. So which role describes the property of stem cells? So stem cells are cells that divide continuously to provide other cells that can differentiate. All right. Now is it able to divide by mitosis to produce more stem cells? Yes. So divide by mitosis to produce identical daughter cells. Okay. Now if you look at it, is only D that says no. So D is eliminated. So we are on A, B, C. Let's go to the next one. Able to differentiate into specialized cells. Yeah. So C is eliminated. We have A and B. So stem cells can differentiate to form different types of cells. Okay. They are undifferentiated. Now, able to repair damaged cells. Well, they may not be able to repair damaged cells, but what they can do is that they can actually form cells that can differentiate to repair. But if you look at this, it says able to repair damaged cells. Able to repair damaged cells. Damaged cells are not repaired. Okay? Damaged cells are replaced. They are replaced. So this is wrong. Stem cells don't repair damaged cells. What they do is that they help to provide other cells that replace damaged cells or repair tissues. Tissues are repaired, cells are. So the answer shouldn't be A. The answer is B. Please note that the answer is B. All right. All right. The answer is B, not C. Is B. Is B. Is this one. All right. Not C. Okay. So let's go on. Look at this question. Let's go. Come with me. The mitotic index is a measure of the proportion of cells that are undergoing mitosis in an area of tissue. It is calculated. All right. So, um, let me take it again. Mitotic index is a measure of the proportion of cells that are undergoing mitosis in an area of tissue. All right. It is calculated using the formula shown. That's the formula for mitotic index. If you don't know it, this is the formula, guys. Number of cells undergoing mitosis all over total number of cells, because it's not all cells undergoing mitosis at the same time. Some are in their interphase, some are in their cytokinesis, but not everyone is undergoing. So this is how you know the cells undergoing in mitosis. All right. Number of cells undergoing all over total number of cells times 100. Now that's it. See, a scientist calculated the mitotic index area of onion root at different distances from the tip of the root. The results are shown. Okay. Now this is the result there. All right. So which statement is correct? Yeah. So which statement? Pause this video and try it. Try this thing. Okay. Which statement is correct? Which statement is correct? All right. Let's go. Remember, guys, as you go into this, we have to remember that this is root tip. If you remember root tip, please. Pardon me. I want to write on this. If this is roots, okay, this is a tip, maybe very tender, and then from here, stuff like this, that's root tip, and then you have all those root hair cells and all that. So you see, root tip. Now this was supposed to be pointed, right? So at the root tip, okay, you see that, yeah, that's the root tip. Okay, just root tip from here up. Okay, this is root tip. Okay, so then I said, which statement is correct? Remember that mitosis occurs highest at the root tip. Okay, so A says no cell division occurs further than 1.4. So we measured from here above. Okay, so away from the root tip, this root tip, so measured at each part, mitotic index was determined, and then move to the next part away from the root tip. Okay, so now, but A says no cell division occurs further than 1.4. So if this is 1.4, it's telling us above 1.4, there is no other cell division from the root tip of the root. from the root tip of the root. Okay, all right, look at it. This is 1.4. Can you see at 1.4 the mitotic index is 2, so that means mitosis occurred here. Now even at 1.6 it still occurred, at 2 is still occurred, so A is wrong. Please, A is wrong because mitotic index occurred all through to 2 millimeters. Okay, it never stopped, so A cannot be the answer. Now B says the rate of cell division decreases as the distance from the root tips decreases. Okay, now you can see that the rate of the rate of cell division decreases as distance from the root tip. Now this is the only thing that can work here, guys. If we had this graph moving this way, okay, moving from here down, you see, towards the root tip, that may work. Okay, but the graph we have is from root tip. So this is the graph we have, distance away from the root tip, distance away from the root tip. So you can see that this doesn't work. Okay, that the rate of cell division decreases as the distance from the root tip decreases. So if the distance decreases, can you see that as the distance decreases from 2 to 1.4, okay, from 1.4 to 2.0 are the same, but check, guys, from 1.4 to 1.2 you can see that it's increasing. As distance is decreasing, right, as distance from the root tip, okay, decreases, rate of cell division increases. Okay, so they have negative correlation. As distance decreases, that means as you come from here down, as you're moving from up here down, the distance, this is the root tip, as distance from root tip decreases, rate of cell division increases because cells divide more at the tip. Can you see it? So you see as 0.2 has highest mitotic index 12. Okay, so if you move away, so if you come closer, the cell division will increase, do not decrease. So B is wrong. C says most of the cells undergoing cell division are close to the tip of the root. Yes, this makes sense. Most of them are close to the tip of the root. Okay, now these ones are undergoing, so there are two millimeters to the root tip, which is here, right? From here somehow, maybe from here 2 millimeters, maybe from somewhere here. Okay, you see that they are the ones undergoing cell division, right? So C looks correct. What do you think? This is for a sample of 200 cells, 0.2 millimeters from the tip of the root. 6 will be undergoing mitosis, but that's not correct. If let's find out what that is correct. If you do, they say 6 out of 6, okay, out of 200. That's 6 undergoing mitotic cells, mitosis or cell division. Multiply whatever you have here by 1,000 by 100. You see that it will give you 3. Try it. Okay, it will give you 3. And they say that this is happening at 0.2. So if you check 0.2, right, is actually 12. Okay, it's 12. So this will give us 3. So the answer is C. Okay, the answer is C. So let's go to the next question. Number 3, which molecule makes up the structure of ATP? This is very important. Check adenine, thymine, deoxyribose, phosphorylates. So which one is the answer, guys? ATP is adenosine triphosphate. So adenine is there, 1 is there. So which options has 1? Okay, A, B, C. Triphosphate, phosphate is there. So 4. Okay, B and C has 4. Ribose is part of. Now ribose is the pentose sugar of ATP. It's not deoxyribose. Okay, so ATP contains adenine. It contains 3 phosphate groups, and it contains 1 ribose sugar. So it is 1, 4, 5. Check it out. So I want to hear your comments. Okay, let's go to the next one. C is the answer. All right, number 4, a short piece of DNA, 19 base pairs long, was analyzed to find the number of nucleotide bases in each of the polynucleotide strands. Some of the results are shown. So this is it. Okay, so how many cytosines were in strand 1? Are you ready? Pause this video and calculate it. All right, pause it. How many are in strand 1? Okay, let's go. Let's go. Pause this video and try to solve it. Okay, so we have, of course, let me use this part here. This is 2 strands. Okay, the first strand and the second strand. This is, oh, sorry about that. Okay, let me use here. Okay, let me use it. This is strand 1, that's strand 2. Okay, so this is strand 1. Let's do it. Strand 2. Okay, so if I should label them, let me label it. This is maybe this is A, adenine. Okay, this is maybe C, cytosine, and this is T, thymine. Each strand has all the bases. Okay, I'm just trying to use this method to get you start thinking. All right, okay, good. Watch this. Strand 2 also, let me say this is T. Okay, because this pairs with this. This is going to be G here. All right, okay, this is going to be T goes with A. That's A here. Okay, and this goes with C. Okay, yeah. So now let's get into the game, guys. So if you check, here, guys, look, in strand 1, okay, we had 4 thymine. Let's look at it. 4 thymine. So strand 1 is strand 1. So we have 4 thymine here. So let me write 4 here. Write it with me. 4 here. Okay. So if you have 4 thymine here, that means I'm going to have 4 adenine, because both of them are complementary. So this is going to be 4. Okay. Good. So adenine is 4. Okay. All right. So quickly, body C strand 2 has 7 cytosine and 4 and 5 thymine. Okay. Now let's go. So let's go to strand 2. Strand 2 has how many thymine? Strand 2 has 4. All right, thymine. Okay, that means it has 4 here. All right, it's going to have also 4 adenine here because they are both complementary. Is that okay? Yep. And then it has 7 cytosine. Strand 2, 7 cytosine. Okay, now this is 7. Now this is 7 here. Okay, so if 7 cytosine is there, that means we're going to have 7. Okay, guanine there. Okay. Yep. All right. So then I say how many cytosines were in strand 1? Okay, guys, watch this. Remember, we had 19 base pairs. How many base? Let's calculate all the base pairs we have here. This is okay, 4 base pairs. 4 plus another 4, 8 plus 7. 8 plus 7 is what? Is 16, right? So this is 4. Yeah. This is 4 base pair plus another 4 base pair plus another 7. Okay, 7 base pairs. So that gives you what? That gives you 16. Check it. 4 plus 4. Okay, plus 7. Right, gives you 16. So the remaining 3 out of 19 will be guanine and cytosine. Is that okay? So remember that we were told that we had 19 base pairs. 19. Okay, so 19 minus 16. Right, 19 minus 16 will give us remaining 1. Minus 16 gives us 3. Okay, so this is 3. The answer should be 3. Do you get? Pause this video and then check it again. All right. So, which statement about the role of DNA polymerase in the process of semi-concentric replication of DNA is correct? Okay, DNA polymerase. Yeah, good. So, guys, we're talking about DNA polymerase. So, DNA polymerase forms the hydrogen bond between complementary base pairs. It does not form the hydrogen bond between them, guys. It doesn't form them. Remember that when this unwinds, okay, when then it forms replication effect like this, kind of, okay. All right, sorry. Here it unwinds, okay. Yeah, see, replication effect. So this is 3 prime, all right, to 5 prime, always up. So this is direction of replication, okay. And this is, of course, this is going to end up, I think, 5 prime here, yeah, sure. So this is the 5 prime end here. That's 5 prime end here. Sorry for my low voice. Okay. So this strand, okay, is used to form the leading strand. So you see, the application copies these two with template strand, but the template strand are copied in 5 prime to 3 prime to 5 5 prime direction. So that means that if I want to make this one the leading strand, I'll copy. So this is going to start with 5 prime. Is that okay? So I'm going to make this, and it's going to move towards 3 prime. Is that okay? This is 3 prime, 3 prime. So if I'm going to copy this one also, I'm going to start from 3 prime end here. This is discontinuous, all right? Discontinuous. I have short fragments, all right? And that's Okazaki fragment. So I'm also, if I'm copying the 3 prime here, that means I'm going to make 5 prime. The new one starts from 5 prime, okay? So what I'm trying to say is that, guys, watch this. This is a DNA polymerase moves along the lagging strand in the 3 prime to 5 prime direction. The lagging strand is a prime, this strand made using 5 prime of the template strand. This 5 prime to 3 prime of template strand. So the lagging strand is this one now, that's made out of Okazaki fragment, okay? If you are making any new DNA, the newly synthesized DNA molecules are made, 5 prime to 3 prime. They are not made 3 prime to 5 prime. But the template is copied from 3 prime to 5 prime. Template is copied 3 prime to 5 prime. Okay? Newly made DNA and strand, which is lagging or leading, they are right. They are made in the 5 prime to 3 prime direction. 5 prime. So this B is wrong. This talks about the template. Template is moved from 3 prime to 5 prime. But look at this. DNA polymerase join new bases to the leading strands. It doesn't join new bases. Bases formed by complementary base pairing hydrogen bonds. Okay, this says that DNA polymerase moves along the leading and lagging strands in the 5 prime and 3 prime. Yes, so this is the leading strand. So it moves in this direction. Okay, but it moves the template strand 3 prime to 5. But in the newly synthesis, this is the leading strand made with this. Okay, 5 prime to 3 prime. Now this second one is a lagging strand, so it's also made from 5 prime to 3 prime. So both of them are made 5 prime to 3 prime direction. Okay, so that is why D is the answer. Pause this video, watch it again, send us your comment. The diagram shows the process of translation occurring at ribose. Yeah, so what is the base sequence at S? So please try this. This is S, this is anticodon. Anticodon, right? This is C, this will go with G, right? Because G pairs with C, is that okay? So this is G, is that okay? Yeah. U goes with A, all right? And then A goes with U. So that's Gaio, okay? The answer is C, okay? Chip. All right, look at the next one. Which row correctly explains how the structures of phlegm sieve tube element and xylem vessel element are related to their functions as transport systems? So they are used as transport systems, okay? So let's see. Now let's check this. Now A says talking about phlegm sieve tube elements contain mitochondria, okay? Contain mitochondria to make ATP for the loading, RT loading. Now phlegm sieve tube does not have the mitochondria that you use for the loading of sucrose is found in a companion cell, all right, not in the phlegm tube element, okay? It's found in the companion cell, this is mitochondria, which provides ATP for loading of sucrose, and that aspect is just pumping out hydrogen ion. Is that okay? All right, so this is wrong. Let's go to the next one. Contains no nucleus, yes, so that dissolved solids can easily move. So this is right. Let's check xylem vessel element on this. End walls between cells are, no, they don't have perforated end walls. They have no end walls. Xylem vessel element don't have end walls. Please note that. They don't have end walls. Did you get? Yes. So C says end walls between cells are perforated, so that, yeah, this is correct. They have end walls, which is called sieve plate. Yeah. Now, lignin in cell walls gives strength that xylem vessels do not collapse due to transpiration failure. This is correct. All right. This says no end walls. No, they have end walls. So this C is the answer. All right, guys. Number 8. Which component of plants is used by the apoplast pathway as water is moved from soil to xylem? Okay. Now, don't forget that apoplast is a movement. Okay. Water moves from one cell to another cell. This is a cell. Okay. Through the cell wall. Okay. Pardon me. All right. Yeah. So it's moving from one cell to another cell wall. So that's apoplast. I'm not showing the other one, so just apoplast. Okay, if it's symplast, is from one cell to another through plasmodium matter, but it's apoplast. Okay, so if it's going to move from this cell to that cell, okay, it's going to move through the cell wall by apoplast, and the cell wall has cellulose. Cellulose is polar, so cellulose forms hydrogen bonds with water adhesion to allow water to pass through the cell wall. So cellulose is the reason why apoplast is possible. All right, the answer is cellulose B. So number 9, which statement correctly describes transport pathways in dicotyledonous plants? Let's see. Okay, so pause this video and try it. So it says in the simplest pathway, water may move through the intercellular space. No, water does not move through the intercellular space in simplest. Okay, it moves through the plasmodium matter. Okay, now I don't think 1 should be there. Now 2 says the simplest pathway may be blocked by the tonoplast. No, this is wrong. It's not blocked by tonoplast. Tonoplast is the membrane of vacuole and is found at the center. So number 3 says in the apoplast pathway, water does not move through plasmodium matter. Yeah, this is correct. Water moves through the cell wall. Okay, apoplast is cell wall. Plasmodium matter is for simplast. So this is correct. All right. 4 says the apoplast pathway may be blocked by Casparian strip. Yes, Casparian strip blocks it at the endodermis. So 3 and 4 looks like the answer. Okay, that's the answer. Pause the video, check it again. Let's look at number. Plants such as tobacco plants retain very little of the water they take in, and the volume of water lost during transpiration is very high. This is because these plants can only absorb carbon dioxide through open stomata. Okay, now you can see plants use carbon dioxide to synthesize glucose molecules. It is estimated that 400 molecules of water are lost for each carbon dioxide molecule gained. So 400 molecules of water equals 1 carbon dioxide. That's a lot. That's a heavy loss, man. How can we 400 for anyway? CO2 is going to be used to make our sugar, so we can eat our burger. All right, 400 to 1. 400 of water to 1 of CO2. All right, let's see how many water molecules are lost for a plant to synthesize 1 molecule of glucose. This is very simple. Tell me the answer. Okay, so if 400 water goes for 1 carbon, the question is how many carbons are in glucose? Do you remember glucose? Hexose sugar, 6 carbons. C6, okay, H12, O6. So it has 6. So if 400, okay, 1 carbon gives us 1 CO2, gives us 400 water. Is that okay? Pardon my writing. Okay, 400. Yeah. Okay, it's 400. So 6 will give us what? 6 carbon. 6 carbon dioxide will give us what? 6 times 400. Okay, which is 240. Yep, that's proportional in math. So thank you guys. The answer is D. So thank you. Please kindly leave your comments. Tell us what you think. Share the videos. So we're going to be doing series 4 later. All right. Take care. Bye.